Good morning, everybody. Everybody, good morning. Welcome to the SECU Daily Planet Theater. This is our globe-shaped multimedia theater. But hello up there on the second floor. Can you hear me okay? Can I ask you, did you see the giant globe outside?
Yeah.
Did you know that this is the inside of it?
Yeah.
Yeah, I kind of figured you did. You seem kind of smart. But it's pretty cool, huh? Not every giant globe has a theater inside, but this one does. And right now we're actually going to introduce you to a scientist that works here in North Carolina, and she studies the water quality. Obviously very important. Water is important to us, essential to life, and making sure that the water quality is good and healthy for us and all the things that live in the water that we depend on. So she is from the U.S. Fish and Wildlife Service, and she's a biologist specializing in water. And she'll give a relatively short talk, and then she would be happy to answer any questions you have. How about a nice round of applause for Michelle Moorman?
Thanks, Brian. So good morning, everybody. It's pretty cool to be here talking in the globe because does anybody know how much of the Earth is covered by water? Yeah, up there. Yeah, 71%. Great job. So, you know, obviously, so much of our Earth is covered by water, that water quality is important. So hopefully today what we're going to do is introduce you to the exciting world of water quality science by telling you about how we measure water quality in our lakes and estuaries, and then talk about a couple studies that we've done here in North Carolina and present some of the stories we've learned from those studies. So just to start with, I love this map. How many people here are familiar with the Albemarle-Pamico Sound? Well, it's a big sound on the coast of North Carolina, and it's actually the second largest estuary in the United States. But I love this historic map because it kind of gives you a great view of how people thought our sound looked like when they first arrived. Is there anything anybody notices from this map? Like just looking at this map, is there something you might think would be characteristic of our eastern seaboard here in North Carolina? Anybody? Well, all that shading kind of makes me think that it's probably pretty swampy, right? So we have a lot of low-lying swamplands in North Carolina. But we've also altered the landscape of our coastal plain by digging a lot of ditches. This one here happens to be the Dismal Swamp Canal, and it was built for transportation, but there's a whole series of ditches in our coastal plain that have allowed us to drain the landscape and use it for things other than swamps, right? So a lot of people go to the coast of North Carolina to participate in recreational activities like fishing, and we have a huge commercial fishing industry in North Carolina that depends on healthy water quality, right? And then we have a lot of small coastal towns. This one's Elizabeth City that border our estuaries, so they have provide an urban landscape adjacent to the estuary. And then finally, this is a picture of some agricultural land in eastern North Carolina. And if anybody's ever driven to the coast, you've probably noticed that we have a lot of farming in eastern North Carolina. And a lot of those ditches have allowed the land to be trained and turned into agriculture. So what does this mean? Why would we want to monitor water quality in our coast and estuaries? Well, there's a lot of reasons we want to monitor water quality. We want to know how our activities on the landscape, right, are impacting the lakes and estuaries in our coast. And so one reason we might want to monitor water quality is just to have a general idea of what's out there, right? What's in the water, some baseline information. Another reason we might want to monitor is to know the condition of the estuary, right? Is it healthy for people and for the fish and other critters that we want to live in the water? And then finally, we actually have common standards that we can use to assess whether we believe water quality is healthy or not. And these standards actually are changing all the time. In fact, we're working on our nutrient standards here in North Carolina right now. So some things to keep in mind when we're thinking about water quality is that there's a lot of natural processes that impact water quality, right? Things on the landscape and climate that impact what is going on in our water bodies. So we have to have an idea of what the baseline conditions are, right? We expect trout streams in the mountains to be clear, but we might not expect that same clarity in our coastal swamps. But one thing we're also concerned about when measuring water quality is pollution, right? We want to know how our activities on the landscape might be impacting our water quality. And if there are things we can do to actually make water quality better, right? So pollution causes an increase, I like this definition, so I'm going to read it to you, in the level of naturally occurring or introduced substances into the aquatic system at a level that is harmful to human or aquatic health, right? We want to make sure we're not creating water bodies that are harmful to us or the critters that depend on them. So a couple basic, things we might be concerned about, right? We monitor for nutrients. So nutrients are like nitrogen and phosphorus. We apply them to our grass and the means of fertilizer. We apply them to our agricultural fields. They're on the landscape everywhere. And it's actually, when you have too many nutrients, it causes a problem called eutrophication. And essentially what that just means is that you're overfeeding your lakes and estuaries, right? Which results in harmful algal blooms. So you see this chart right here, increasing nutrients cause an increase in chlorophyll A, which is a measure of how much phytoplankton might be in the water. And so, although we want phytoplankton there, they're the base of our food web, we don't want too much phytoplankton because it can cause harmful effects in our ecosystems. In addition, we're concerned about too much sediment in our waters. That can change the habitat, it can change the bottom habitat and the fish that maybe depend on more gravelly habitat to survive. In addition, it makes the water more turbid, which can impact water clarity and impact the ability for plants and animals to survive. And so as you can see here, on the left is a lake that's turbid and nutrient enriched and dominated by phytoplankton. And on the right is a healthy bed of submerged aquatic vegetation, which we like because submerged aquatic vegetation is good for fish and it's good for birds. But poor habitat and water quality can damage aquatic ecosystems. So that's one reason we want to measure water quality. Another concern we might have is contaminants in the ecosystem. So this right here is a diagram of the mercury cycle. But too much mercury or other contaminants in the water can have harmful effects on human and ecological health. So there's a lot of reason, just like we go to the doctor for our yearly physicals, that we want to go out and we want to monitor our water bodies to make sure that they're healthy. And if they're not healthy, we might want to take some actions to try to improve the health of that water body. So pollution can cause healthy ecosystems to provide fewer ecosystem services. Have people heard that term before, ecosystem services? It's pretty popular buzzword nowadays. But ecosystem services are the things that we want our environment to provide. So we want our environment to provide healthy fish and great recreation opportunities and clean drinking water. And so we want to not pollute so we can continue to receive these ecosystem services. So what's my role as a scientist? The name of this talk is Every Drop Counts, right? So I'm just one drop in the realm of scientists working on water quality issues in this state and in this country. And it really is a team effort. So we all have to work together because it takes a lot of work to figure out what's going on in our lakes and estuaries. But as scientists, we work to identify environmental problems and issues, and then we collect water quality data and we interpret it and analyze it, and then we share these findings with the public. So that's our job. And we use the scientific method. Most people here are probably familiar with the scientific method. So, right, we can't answer every question. So we have to decide what questions are important. And then by determining our monitoring and research objectives, we then have to develop measurable research questions. So for instance, our state does a great job monitoring our sounds. They have a long-term monitoring program in the Albemarle-Pamico Sound. They've been going back to the same sites since the 1980s. It's a really important program because they're interested in long-term trends in water quality. And without that data, going all the way back to the 80s, we wouldn't actually know if the water quality in our sounds was improving or declining. And so that's the research question. What is the health of our sounds? And then once we come up with that research question, we have to develop field methods and take measurements, right? We got to go out and we got to do the work and collect the data, or else we won't have any data to analyze. And then finally, once we've analyzed the data, we discuss our results. So just briefly, how many people, has anybody here ever taken a water quality sample? A couple people. Yeah, so it's pretty fun, right? You get to go in the field, you get to hang out on the water, you get all this fancy instrumentation. You collect some data, you write it down, you come back. It's exciting stuff. And so as water quality scientists, there's a lot of measurements we can take in the field, right? We have all these special instruments that we can go out and we can measure in situ temperature and pH and specific conductance and turbidity and biological communities, and we can learn something about current conditions in the water body. This right here actually is a continuous water quality monitoring station. So as our technology's gotten better, we've been able to do more. But this station actually has a probe that's deployed that takes measurements real time, and we can go online and see what's going on in the lake without even going out there. This is pretty nice if you don't feel like going out on a boat in a rainy, cold day. Now, some things we can't measure in the field. They're a little bit more complicated. They take complicated instrumentation to analyze the chemistry. And so we actually have to go in the field and collect a sample of water and preserve it. So this right here is actually a mobile lab that we use to process samples. And then once we process those samples and preserve them, we have to throw them in a cooler and ship them via FedEx to a laboratory that can actually do the analysis. So, this parcel of water might travel a long way to just return us one number. It's kind of exciting when you think about it. Some of this water is better traveled than me. But so all this data, what do we do with it, right? That's like every scientist's question and problem. But we got to go in the field and we got to collect the data. And then we have to get our data back from the lab and we have to track all this data. And then finally, we have to sit in the office and make sure that the data is correct and everything's good. And then after we've done all that work, we can finally actually analyze the data. So it takes a long time to get to the point where we can actually say something about water quality. And once we've done that data analysis, we can then share our results with the public. Because you guys are the ones that want to know what's going on with our sounds and estuaries and lakes, right? So just briefly, I'm going to talk about 2 water quality monitoring programs I've been involved in. And hopefully this will give you guys a better idea of kind of what we do as water quality scientists and what we learn and why that information is important. So I am a biologist with the Fish and Wildlife Service, and I currently work at Lake Mattamuskeet National Wildlife Refuge. So Lake Mattamuskeet is a large lake. Here's a small map of North Carolina, and you can see Lake Mattamuskeet is located out here on the Albemarle Peninsula. It's a 40,000 acre lake, and it's one of the most important habitats for wintering waterfowl in North Carolina. So Lake Mattamuskeet, one of the reasons it's such an important habitat for waterfowl is it did at one point in time contain these dense beds of submerged aquatic vegetation. So this is the second time you've heard about submerged aquatic vegetation today, which probably makes you think submerged aquatic vegetation, good, right? Good water quality. And it's something that we want that's indicative of a healthy ecosystem here in North Carolina. Well, a major management concern at Lake Mattamuskeet is that this submerged aquatic vegetation, which you might hear referred to as SAV on occasion, has declined, right? This is a picture from 2012. And on the west side of the lake, we see that turbid, nutrient-enriched, phytoplankton-dominated environment. And on the east side, we see a clear lake full of SAV, right? And so when we started doing water quality monitoring at this lake back in 2012, people really didn't know why the SAV had disappeared. We had all these hypotheses, but we didn't have any information or data to really be able to say, hey, what's wrong with the lake? People just were concerned. And so what we found the last few years, we've been doing these biological surveys, right? So these are things we can just go out in the field and we can run transects and we can say, hey, there's SAV here, submerged aquatic vegetation here, or there's not, right? And so back in 2013, right, you see there's not a lot of vegetation on the west side of the lake. But there's actually the east side of the lake, looking at these pie charts, right? So west side of the lake, 87% bare. East side of the lake, 62% wild celery, which is a type of vegetation. We're like, east side's pretty healthy. West side's not, right? And then the next year, whoa, what's going on here? Both sides of the lake, a lot less vegetation, right? So we're like, what's going on in the lake? Why is the vegetation disappearing? So this is our biological monitoring, right? We know that there's something going on in the ecosystem that's not good. So we're going to do some other measurements, right? We're going to do some real-time measurements to get a better idea of what is going on in this lake on a daily basis that might be causing the vegetation to decline. So this right here is some real-time data from the USGS, which has been one of the partners on the project. I should mention another partner has been the Division of Water Resources. They've been running our samples at the lab, as well as Fish and Wildlife Service and Wildlife Resources Commission. But pH is a measure of how basic or acidic a sample of water is. And one reason we worry about pH in North Carolina, and we have a state standard, so the state standard is actually we want pH between 6 and 9. Now, the 9 standard is actually the reason we have that standard is when pH gets too high, it's usually an indication that a phytoplankton bloom's going on, right? So here we can see where that state standard of 9 is being exceeded on a daily basis in the lake. So this might be indicative that we have phytoplankton blooms occurring in the lake. And so that's where this real-time data, this data that's being collected on a sensor and transmitted to a satellite, and then it comes back and we can go see it on the web, is really valuable because we're like, wow, every afternoon the phytoplankton, they are producing, they are blooming, and we have these phytoplankton blooms going on indicative of these peaks of pH each day. So we're like, well, here's one thought of what's going on wrong with the lake, right? There's probably a lot of phytoplankton in the lake. And that's causing poor water clarity, which might be causing the SAV to decline. So some other things that we can do is actually, take some additional samples. This is where we go out and we collect that parcel of water, right? And we see what's in it. And we can use this information to have a better understanding of how many nutrients are in the water or what other types of contaminants might be in the water. So we don't get this information on a 15-minute interval, right? We don't have these nice, very full data sets. But what we can do is use this information and look at trends through time. So they've been actually working, the state has been taking samples on this lake since 1980s as part of their lakes assessment program. Now these samples are pretty sparse and sporadic, right? So they only happen every 5 to 10 years. And then when we started doing the real-time monitoring in 2012, we started collecting samples on a monthly basis, which explains why we have so much more data there. But it's enough data to see that there's a significant increase in chlorophyll A through time, right? And chlorophyll A, for those of you guys that don't remember, is an indication of how much phytoplankton might be in the water column. So remember back to that thought that, we like phytoplankton, phytoplankton's good, but too much phytoplankton is kind of like too much of a good thing. It's kind of like, when you eat too much, that's maybe too much of a good thing too. So our state actually has a standard for chlorophyll A because they realize too much phytoplankton's bad. That standard's 40 micrograms per liter. And you can see, now this has all been log transformed, but, this one hasn't actually, sorry. That 40 micrograms per liter standard since 2012 has been exceeded, and the blue is the east side of the lake. So I don't know if you remember, but originally there was healthy submerged aquatic vegetation communities on the east side of the lake. And then they've declined in the last 3 years. And you can see Back in 2012, chlorophyll A was below that state standard of 40 micrograms per liter. And just in the past 3 years, we've seen that both the east and west side of the lake continuously exceed that chlorophyll A standard. And so we can also look at what's been going on with nutrients through time in the lake. So again, we're looking all the way back to the 1980s when the state started monitoring the lake. And we're looking at total phosphorus here. And we see that there's been a significant increase in the amount of nutrients in the lake during this time. And so we can also look at nitrogen, another nutrient that can cause harmful algal blooms. And again, going back to the 1980s, we can see that there's been a significant increase in the amount of nutrients in the lake through time, the amount of nitrogen. And so what do we do about it, right? Did all that nutrients come in overnight? Anybody have any guesses? No, they've been slowly added to the lake through time. So the next question is, how can we improve water quality in the lake to restore the ecosystem, right? How many people think here that we can change the lake overnight? Well, that's good. Glad to hear that, right? It took a long time to get all those nutrients in, so it's going to take some time for them to be reduced and restore the ecosystem. And during that time, we will, have to continue to monitor to see if our restoration activities are successful or not. So that's kind of one example of how data through time can help us better understand our ecosystems. I'm just briefly going to talk about another project we had where we were monitoring the Albemarle Sound. And this was a project where we actually did a snapshot in time. And we went out and we collected a lot of samples, kind of like AppNep's doing this summer, to see what was the condition of that ecosystem through space, right? So one summer, collecting a lot of samples to get a better understanding of water quality in the Albemarle Sound in the summer of 2012. And so, just briefly, this was kind of a baseline study to get a better understanding of, what were the nutrient conditions in the sound and were there harmful algal blooms occurring. And so you can see we go out, we visited 40 plus sites, right? And we can get all that data back. And then what we're looking at here is just we take that information and we make some box plots. Really simple stuff that tell us about the distribution of our data. So here we have chlorophyll A. Remember that index of phytoplankton, right? How productive the water is, but it's an index. You're not actually measuring phytoplankton. And then we have cyanobacteria, which is it's actually a bacteria, but we study it with phytoplankton, and it often causes harmful algal blooms. We have the total density of algae and the biovolume of algae. And then these red lines are indicative of state standards or World Health Organization guidelines of whether or not we consider it to be a healthy ecosystem. And so what we can see is we have kind of a eutrophic system in the Albemarle Sound, that the levels of cyanobacteria and total algae are relatively high compared to these standards and guidelines. And that was just in the summer of 2012, going out collecting samples throughout the sound. So we can say in 2012, the level of cyanobacteria in the Albemarle Sound was kind of high. That's what we can say. We can also take that information and we can put it on a map, right? People love maps. How many people here love maps? I love maps, right? So one thing we do when we go out and we collect a lot of information spatially is we put dots on the map, right? This is a very favorite exercise of scientists. But what it allows us to see is the distribution. Here we're looking at the distribution of cyanobacteria. And so the red dots are samples that had cyanobacteria cell counts of greater than a million. And we see that, in the northeast portions and the central portion of the sound, those are the portions where we had the highest levels of cyanobacteria. So if we're worried about nutrient pollution and places that harmful algal blooms might be occurring in summer of 2012, these are the areas we might want to look at for further investigation. And then finally, we can take all this data and we can start to make relationships, right? So this is another thing scientists love to do. We love to, look at one parameter versus another. Is there a relationship? And here we're looking at the relationship between nitrogen and chlorophyll A. So going back to the beginning when we said, as nutrients increase in a watershed, we expect that the amount of phytoplankton will increase in the watershed. And we see that's true here in the Sound, in the Albemarle Sound in 2012, that as we had more nitrogen, we had higher levels of chlorophyll A, right? So we can go ahead and we can say, yeah, you know, too much nitrogen might be too much of a good thing. And so, you know, why do we care, right? Well, we want our sounds to be healthy because we all like to go recreate there. We like to eat our fish from there. And we know that excess nutrients can cause harmful algal blooms in our lakes and our estuaries. So we're going out and we're monitoring to see what the condition of these estuaries are, and we're learning about the system. And hopefully this information will help our state make better decisions as they're developing nutrient criteria for our lakes and estuaries currently. And I just thought this was a funny cartoon. Has anybody here heard of Indian River Lagoon? It's a lagoonal estuary in Florida that's gotten a lot of press because it has a lot of harmful algal blooms. And so we don't really want to be Indian River Lagoon, right? So we want to make sure that we don't put too many nutrients into our lakes and estuaries so people aren't writing funny cartoons about us. But just in closing, I want to talk about the benefits of partnerships in the Albemarle-Pamico Sound and in water quality monitoring, because each one of these little drops of monitoring programs that I've been involved in, which is just a small amount of the water quality monitoring that goes on, took a whole group and team of people, right? It wasn't just me going out and figuring out all this stuff. It was, you know, other staff and interns and other agencies all working together to get our samples, to get them analyzed at the lab, to bring the data back, to analyze it, to just get a little bit better understanding of the processes going on and the status of our lakes and estuaries. So it really takes a team effort in order to be successful at water quality monitoring here in North Carolina. And you know, that's where umbrella organizations like the Albemarle-Pamico National Estuary Partnership, I think they changed their name now, help us all kind of work together and bring together the different people working in the sounds and the estuaries so we can all get a better understanding of what is going on with water quality. And so through partnerships, we can improve our scientific understanding of the health of our lakes and estuaries, and that's really kind of an important take-home message. So, you know, be sure to support your water quality monitoring groups and agencies. They do really good work. I hope you just got a flavor of it today. And with that, I'm happy to take any questions.
How about a nice round of applause for Michelle Moorman from the U.S. Fish and Wildlife Service, and wasting no time at all, we have a question.
Can Madam Muskie be saved?
That's a good, I think save is the wrong word, right? Repaired. There are examples of other small, shallow eutrophic lakes in the world that have been restored. You know, it's probably a twofold issue. One is we have to reduce the amount of nutrients in the lake. And then the second is actually, you know, undertaking some restoration practices that might jumpstart or kickstart submerged aquatic vegetation growth.
Is that from surrounding farms?
That's probably one of the contributing inputs. There's a, you know, we haven't actually, I guess, identified specifically where the nutrient inputs are coming from, but there is a lot of agricultural land that does drain into the lake. So I'm sure that's one of, you know, the land practices contributing nutrients to the lake.
Any other questions? Yes.
You mentioned that you haven't looked at identifying the sources yet, but as a rule of thumb, the east side versus the west side, is there land use differences that you can maybe look to for why one side is worse off than the other?
Potentially, there's some, I guess, policies and regulations in place that allow for pumping to occur directly. So, you know, down at Lake Matamesquite, it's pretty low-lying land, and so there's an extensive network of drainages and canals that have allowed the land to be farmable. And some farmers actually, due to some policy and legislation put in place back when the lake was turned into a refuge in the 1930s, are allowed to pump directly into the lake. And so and that's on the west side of the lake. So that's possible maybe that the west side of the lake kind of showed symptoms of eutrophication first because they were receiving these kind of direct, maybe higher levels of nutrients because of the actual active pumping into the lake. Mm-hmm.
I have sort of a twofold question here.
Mm-hmm.
Is there any concern or consideration of the waterfowl that migrate there annually and their residue that they might leave? And also the second part would be, is there any movement of the water either down deep or on the surface caused by winds or other actions that might influence this?
Well, those are two great questions. So the first having to do with, I guess, nutrient inputs from waterfowl, which is what you're asking about. So another potential source of nutrients to the lake might be there's a lot of waterfowl impoundments that surround the lake because it's a very popular area for waterfowl hunting. And so in February, these waterfowl impoundments are on farm fields surrounding the lake, and the farmers need to drain their farm fields in order to be able to grow crops. And so it is possible that that might be another source of nutrients, but it hasn't been studied yet. And so that's why, I mean, we have an idea of what the sources of nutrients are, but we don't really know how much of a contribution might come from, waterfowl impoundments versus, from the farm fields directly versus atmospheric sources versus in situ recycling of nutrients in the lake, right? All these nutrients have kind of been added to the lake over time. And it might just be that it's this self-feeding system where nutrients are constantly recycled within the lake. And then to answer your second question, it's a very shallow wind-driven system. So there's a lot of wind. Any of you have ever been to the Outer Banks? Matamesquite's not far from the Outer Banks. And so, it blows pretty regularly out there, which is kind of nice if you're a windsurfer like me. But so this constantly is churning up the lake. And we've talked about how, it's kind of this negative feedback loop. Like you have these phytoplankton blooms, the submerged aquatic vegetation has declined, so they're no longer there to kind of hold the substrate together. You get these wind events. So another source of the turbidity might just be suspended sediment that's getting resuspended during these wind events.
I know this is sort of a longer-term gradual thing, but climate change, does warmth also mean something like there will be more algae and more, does rising temperatures mean, is that also problematic?
Well, rising temperatures will change probably. Different phytoplankton have different kind of temperature preferences. So, as water temperature changes, as you have a longer growing season, that might actually change what type of phytoplankton are living in a lake or estuary, and it changed the salinity regime, which some phytoplankton, prefer fresh water versus more saline environments. So, yeah, climate change is going to change our ecosystem as well, and that's, the next challenge is, well, the first challenge is just kind of figure out what's going on now, and now we also have to throw in climate change and kind of figure out how that's going to change the ecosystem too.
So I know that you said there's a lot of unanswered questions about what the actual sources are, but is there any, even just knowing what you already know, are there any kinds of recommendations for people, for farmers, are there any kind of general things that we already have to pull back on or change?
Well, I think that's where we have to kind of consult with our, agricultural engineers who have specialized in, kind of working with coming up with restoration practices on farms to help reduce that nutrient loading. So, you know, as water quality scientists, we probably have the best job, right? We go out, we measure, we make statements about what's going on, you know, in a watershed. But then we have to rely heavily on kind of our ag engineers and, you know, the people that are out there engineering solutions on the landscape. Well, just like here in Raleigh, you know, we have best management practices for stormwater runoff. So the same kind of thing, we need to rely on people who can really help us better understand, you know, what kind of BMPs could be put in place to make an effective change in the lake.
BMPs?
Oh, sorry.
Management
Practices. I know, bad jargon, sorry. I tried to avoid it.
Any other questions before we let the show go? Well, how about this? So is that you? Is that a picture of you?
Oh, yeah.
Ah, nice. It's good to see the other side of scientists. We have scientists here who ride unicycles, and we have who knows what else. And it's good to see when people think scientists are just old men with glasses that work in a laboratory, and it's like, no, this is what scientists are.
Right, and then it's another reason, right, we care. Because if I'm going to be out recreating on the water, I want it to be safe and healthy.
Selfish reasons. You want to be able to windsurf and stuff.
Not purely selfish.
No, but why did you get into this field? Why did you choose this path and water? You're a biologist. How did you focus on water and water quality? How did you come to that?
Well, that's kind of a good question. I guess I always grew up loving to play in like the streams and lakes and loved being around water. And so I actually started after undergrad with the USGS as a hydrologic technician. And, you know, I was like, oh my God, I'm getting paid to go out and hang out in the streams and collect bugs and fish. Like, this is so cool, right? And then after you do that for a while, you kind of decide, wow, I want to know more. Like, I want to know what's going on with the environment. And I want to better understand how I can interpret this data I'm collecting. So I went back to grad school and, you know, improved some of my skills. And that's kind of how I ended up down the water path.
So, any other questions for Michelle before we cut her loose? All right, how about a nice round of applause for Michelle from the U.S. Fish and Wildlife Service? Thank you very much, Michelle, and thank you all, and thanks for the questions, and enjoy the rest of your visit here or work day. Thanks.
Thank you.